Thanks very much, Peter. Yes, I'm going to be talking about the ENIGMA VBM tool, its development, and some of the results that we had. So this was it was the first VBM study, and it's a nice link because I'm from King's College London. It's linking King's, UCL, and VBM. So it was a study on 15 patients with schizophrenia. It was a collaboration between UCL, the then Institute of Psychiatry, and the MRC Cyclotron unit. So the Institute of Psychiatry has now become the Institute of Psychology, Psychiatry, it's so long I can't say it, Psychiatry, Psychology and Neuroscience at King's College London. I also did my PhD at the MRC Cyclotron unit at Hammersmith Hospital too. So VBM has continued to evolve since this date, as we've had optimized VBM, DARTEL, CAT12, but how about sample size? How has that changed over the time period? So this was a study that we did looking at a meta-analysis in patients with bipolar disorder. And one of the things we plotted here is the year of the study versus the total sample size. So that's patients with bipolar disorder and controls. And one of the things we saw was this sort of flat, you know, all the way from the 1980s till the date of the study, the sample size was flat. And what had happened here was there were type 1 errors. This was due to multiple comparisons within studies where they hadn't properly corrected for. And that actually gave the erroneous impression that effect sizes are large and those sample sizes were adequate because they've been finding all these significant findings and they thought, right, okay, we're fine, we'll keep to the sample size, but those were actually type 1 errors. However, from the meta-analysis, we found that the effect sizes were actually very small. So we also have type 2 errors occurring as well. So we have this whole literature contaminated by type 1 and type 2 errors. So the only way really to resolve this, I think, was to increase sample size. But one of the conclusions we came to in this paper is there had been no change for 25 years. So that was what the situation was, but what happened over time is that we have a study that came out in 2017 with a whopping 6,000 participants. And that was due really to the ENIGMA Consortium. So what is the ENIGMA Consortium? Well, it stands for Enhancing Neuroimaging Genetics through Meta-Analyses. And although the consortium was originally sort of focused on genetics, they've kind of moved on a little bit because even those effect sizes are very, very small. The UK Biobank kind of supplanted that, but a lot of it is looking at case control findings in patients with different psychiatric disorders. And we've got a world map here showing some of the sites involved with the different colors being different disorders. So data sharing has been a real hallmark of ENIGMA, and that all sounds great, but we do have some problems with data sharing. So some sites may not feel comfortable with sharing private patient data. We've got issues of GDPR and also data sharing agreements. So this has been the bane of my life sometimes. So this is a legal agreement between two universities, and every time you need to set up data agreements between each site, there's two sort of legal departments that get involved, so it can really sort of slow things down. So how can we try to expedite data sharing in neuroimaging? Well, one way to do this is to try to work with group data, because this is not identifiable. You don't need to worry necessarily about GDPR in situations if you're sharing group data. So you can have a situation where each site uses the same software, which standardizes their analysis of data and produce statistical group data, which can be sent and meta-analyzed at the coordinating site. So based on this premise, what we decided to do was develop a user-friendly VBM tool for the ENIGMA Consortium, which has limited user intervention. So we try to prevent any heterogeneity in how it's run. It's standardized and includes a comprehensive quality control and produce these statistical T-maps for meta-analyses and only transfers group data. Okay, so here's a schematic of how it works. The software requires nifty images from the patients, nifty images from the controls, and also a standardized text file that the ENIGMA Consortium are already using. So we have the subject ID, the diagnosis, whether they're a patient or not, their age, sex, and any number of clinical variables. That's then all used by the ENIGMA VBM tool. We've tried to make this easy to run with lots of documentation. We run lots of auto quality control and sensitivity analyses, and I'll explain a little bit more about that in the results. And then in terms of what leaves the site, so that's that dotted line, we just have voxel group data and QC data. So no patient level data leaves that site. And that's the T-map. So we would have a T-map of the case control findings, but also perhaps a regression of a clinical variable as well. So we collect those from all the different sites, and then we run a voxel-wise meta-analysis. So each voxel is meta-analyzed separately, and that's using SDM. So SDM stands for seed-based demapping, and it was developed by a researcher called Joaquim Rada while he was at King's College London. He's now moved to Barcelona. And one of the advantages of this tool is it uses robust family-wise error correction for multiple comparisons using threshold-free cluster enhancement. We don't really need to look into this in detail, but this is a kind of overview of how the tool works. We have 2 published papers so far in early onset psychosis and PTSD, so you can have a look at that if you're interested. But the core of the software really is the Dartel processing pipeline. Okay, in terms of results, what have we found? Well, this was the first study published looking at early onset psychosis, and this was led by my PhD student, Shijing Si. And what we had here was, patients with early onset psychosis. So this is psychosis before the age of 18. Usually these are very difficult to recruit, so they're very small samples. But we recruited 15 cohorts with 482 patients, 469 controls. So what did we see? Well, we saw decreased brain matter across the whole of the brain, really, in these patients versus controls. So I should say that we've controlled for age and ICV here. And we've thresholded at, we've actually gone to 0.025 family-wide error corrected because we look at both the increases and the decreases. So brain matter reduction in all of the brain, with the greatest reduction in the left medial cingulate. And I should say that the units for the color there is Hedges G, which is a measure of effect size. If you're not familiar with Hedges G, it's basically exactly the same as Cohen's D. So Cohen's D of about 0.4 is the same as Hedges G of 0.4. But we don't just have to look at case control findings. We can also look at clinical variables. And we were interested in if patients were having more strong antipsychotics, was there a relationship with brain volume here? And this is where we saw reductions in the orbitofrontal cortex with a smaller effect size peaking at 0.2. And this was in the patient group only. And sort of, I guess the take-home message of that is the findings that we see in the case control differences can't simply be explained by medication because they don't cover the entire brain, and they're only in a particular, the Hedges G effect size is a lot smaller. Another kind of key issue, a key way that we use the software is we try and make the data available as we can. So the final 3D effect size maps we upload to Neurology. I don't know if people are familiar with this website, but it's here we have some data from PTSD, and that was run by Cheryl C. So that's available at the moment because that's published. So the advantage here is that data can be downloaded, or you can query it with online software such as Neuroscience. We can also look at the effects of VBM parameters. And so one of the philosophies of this is that we have an a priori set of settings that we use for the main analysis, but we also vary a number of parameters because we're also interested in how VBM performs in different ways. So our a priori is 8mm smoothing, but we can also look at the effects of 12mm and 4mm smoothing. So this is an example looking at obsessive compulsive disorder. This is not published yet, but we had about 2,000 patients with OCD, 2,000 controls, and that was run by my MSc student, Chao Yu. Usually when we look at different disorders, we haven't seen correlations with symptoms, but we have seen this with major depressive disorder. And I think this is probably because of the size of the sample. So in total with the patients in control, we have over 10,000 individuals, people who had a depression data. This is the Beck Depression Inventory. with a high score being with severe depression. We saw most patients with moderate to severe depression. And we saw around the cortex and a lot of the grey matter, actually, very subtle reductions in grey matter volume. And you can see the effect size here is now getting quite small. We're able to pick up these very small effect sizes with these very large groups. That's analysis been done by Kai Huang, who's a present PhD student. We're also looking at bipolar disorder as well. And this is an animation really just to show how we get more confident with the findings as we add more and more site data. And it also shows how international the data is. So you can see that as we add the different samples, these points all around the world. So it's very well represented. So we're not only increasing sample size, but we're getting global representation as well. So what we saw with bipolar disorder, again, grey matter volume, a lot of it affected, potentially more the thalamus. And we saw this characteristic region, the splenium of the corpus callosum. And that's run by Shane Prakash, who's just starting her PhD. I should add also that the color coding here we've used is the Z score rather than the Hedges G. So this is really a measure of significance of how confident we are about the results. Because we've used voxel-wise data, we've got about 200,000 voxels associated with a Hedges G effect size. And what we can do is we can plot a histogram of the Hedges G effect. So what we've got here is a graph of the number of voxels on the y-axis in the normalized and template space, and the Hedges G along the x-axis. So anything from the right-hand side of the 0 line is showing a reduction in grey matter volume. Anything to the left is showing an increase in grey matter volume. And I should say this is non-thresholded data. So this is all the data that's available. We have our peak effect size for early onset psychosis, about 0.54. But you can see that probably the most frequent type of voxels around about effect size of 0.2 in terms of its reduction. So I found that looking at these histograms is quite a helpful way to compare disorders, because we've almost got data overload when we're looking at lots of different disorders. So what you're seeing here now is a number of different disorders. If we start on the right-hand side with the most severe reductions in grey matter volume, we've run this on a relatively small sample of the Albany sample. So the highest, so the most frequent voxel being about 0.5, but it would stretch a lot further wide to the right. We then have schizophrenia as we come to the left, so more established psychosis, early onset psychosis, panic disorder, and then we've got something like obsessive compulsive disorder showing perhaps the smallest change. So when I was looking at this graph, I think my take-home messages from this is, one, in terms of psychiatric disorders, it's a story of reduced grey matter volume. So we really don't see any increases. And those increases that you see on the left of the 0 line, I don't think any of them became out significant under family-wise error correction. Another thing that I was interested in is I don't see a clear dichotomy here of neurological versus psychiatric disorders. So actually, as you move to the right, you kind of see the severity increasing in psychiatric disorders, and there's not a huge gap with Alzheimer's. It's actually a kind of a sort of general curve there. So in terms of conclusions, effect sizes of brain structure in psychiatric disorders are small. So obviously large sample sizes are needed. So that's why we need to go to ENIGMA. Sharing data can be challenging, particularly in the current climate of universities being very cautious about this. So the methods that we use potentially allow more sites to participate. We've used the ENIGMA VBM tool in over 8 disorders and 25,000 individuals, showing robust changes in brain structure. One thing is a word of caution. We still really need to be cautious about unmeasured confounding. So I personally think some of the sort of suspects here are smoking, BMI, alcohol, and movement in scanners. So all of these things could potentially be associated with particular psychiatric disorders and have an impact on brain volume. However, ENIGMA is working to collect more detailed information from sites to examine these effects. So just want to acknowledge none of this work would be possible without the generous collaboration of 150 sites around the world. I'd like to thank my PhD students, people working in ENIGMA and all the working groups, and Carl Christo and John Ashburner we're initially starting VBM. Thanks very much.